Hey, Joseph.
Hey, Andy, how you doing?
Hold on, let me change my audio.
Hey, we're recording this session, just FYI.
Try it again.
I said we're recording this session, just FYI.
Okay.
How you been?
I've been good. It's been a busy week, but doing okay. How about you? How's the family?
Good. Busy.
Yeah.
We have a lot of people joining, so
Okay, good.
we have 25 people signed
Let
up.
me go ahead and try to share my slides.
Yeah. Let's try that. Make sure that's working.
It's always a trick managing all the windows, right?
Absolutely.
All right, so hopefully what you're seeing are the slides and not like the
Yeah.
presenter. Okay.
Yeah, that's what we're saying. Yep. We still have 2 minutes. Usually people start joining at exactly at 11, so we'll give it another minute over 11.
Have you guys been doing any in-person seminars as well, or are they all virtual?
No, that we wanted this seminar series to be online just because we have partners in Europe, and we wanted to be able to host them as well and make it open to everyone.
Gotcha.
So the plan is to keep it online. I don't know if we at some point talked about having, distinguished seminars where, we would host somebody on campus and then broadcast it somehow.
Mhm.
But we didn't discuss it in detail. So far, we're just gonna keep doing this.
Gotcha.
Yeah.
Yeah, I think it's probably going to change how a lot of departments do seminars for the next many years, right? Because it's cheaper and it's
Oh, yeah.
easier to invite people, but it makes a different experience, I guess. So.
Absolutely. I mean, it's always good to host somebody on campus. I mean, that's honestly how I meet a lot of great people. You welcome them in your office, you talk research, you have something to discuss. But I mean, this one here, it's also good because you can reach a lot of people, and that's the point.
Yep.
I don't know. It's I think a balance would be good. So it's 11. I think we should give it another minute or so, and then
Yeah.
hopefully we have already 9 people attending, so we're expecting a lot more. So let's just give it another minute and we can start.
Can you see my laser when I do that?
Yes.
Okay, cool.
I think the rest of the people will join as we proceed, So maybe we can go ahead and get started. Is that good?
Sure. Yeah, I'm ready.
All right, everyone. Hello, and thanks for being here today again. This is our monthly Living Materials Talks, or LIMtalks. Today, we're pleased to welcome Professor Andy Sarles. He's a very special guest from the University of Tennessee. He's very special to me because he's a friend, colleague, and my former postdoc advisor. Dr. Andy Sarles is currently an associate professor in mechanical engineering, in mechanical aerospace and biomedical engineering department at the University of Tennessee. He received his PhD in 2010 in mechanical engineering and a master's in mechanical engineering in 2006, both from Virginia Tech. Today, he will be sharing with us his research on biomimetic interfaces for neuromorphic signal processing of sensory information. This is a special topic that we're interested in the Center of Living Materials. And with that, I would ask you to give your full attention to Dr. Sarles and save your questions until the end of the presentation. Dr. Sarles.
Great. Well, thank you, Joseph. Good morning, everyone. It's great to be here kind of virtually with you. And thank you for the kind introduction. It's always good to see you, Joseph. Thanks for the opportunity to speak with you today about my research at Tennessee. So the title for my talk is Biomimetic Interfaces for Neuromorphic Signal Processing and Sensory Information, where the word neuromorphic, I'm not sure if you've heard of this, refers to hardware or software that are structured to operate in ways that emulate the brain. So I'm going to talk to you a little bit about some of our recent work. So a grand challenge in engineering is the development of intelligent systems that can operate autonomously and learn in real time in unstructured environments. Such a system must be able to perceive its environment, must be able to interpret this information to make decisions, and actuate reliably with control. Traditionally, engineered systems such as robots have been pre-programmed to carry out defined tasks. And so the Boston Dynamics dancing robots are a good example there. They're not learning how to dance, but they're being programmed specific choreography to carry out those motions. But more recently, AI and machine learning techniques are being used to train systems, engineering systems, for doing pattern identification and even decision-making including in robotic systems. And so the Spot dog type robot from Boston Dynamics is a good example of a system that uses AI to process images for doing some pattern identification and decision-making. So this latter approach leverages the mathematical power of neural networks, which are AI and machine learning algorithms, and lots of data. But this takes lots of computing resources and energy to power those computing elements. And it's often performed via cloud computing in large data centers. In contrast, the human brain and its nervous system is the most complex sensory and computing system that exists. Brains are massively parallelized structures consisting of approximately 100 billion neurons, each which connects to another 1,000 neurons. So this interconnectivity forms a dense hierarchy for information processing and storage, and it occurs across roughly 100 trillion, 10 to the 14th, synaptic junctions between neurons. So this scale is necessary in part because the brain must make sense of all the data that's coming in from the different senses and the sensory receptors and neurons that are distributed across the body. So these are our 5 senses, of course, and to give you a sense of density of sensors that are feeding information to the brain, we have about 13,000 mechanosensor receptors on each finger. And so that, you can imagine, provides a huge amount of data that the brain has to make sense of. So while it's not as fast in terms of speed as transistor-based computers, biological brains, which are also soft, squishy, and they operate at room temperature, consume far less energy, and they occupy far less space than modern supercomputers. In fact, this comparison shows that a brain is roughly 5 to 6 orders of magnitude better in computational efficiency and density when you normalize the number of calculations per the volume or per the amount of power spent. So this level of performance really is uniquely enabled by the complex hierarchy that we just looked at in the brain and the use of activity-dependent synaptic plasticity, which can include both functional and structural changes that occur in these synaptic junctions at the molecular level. And so this adaptability, where a synapse can change kind of its state to be more receptive to information flow, depends on the information that's coming to it and how often it receives these cues. And so it's called activity-dependent plasticity because these changes depend on when information is sent to the synapse from the neurons. And importantly, it's used to modulate and store a strength, or sometimes called a weight, of connectivity between neurons. And so by being able to adjust these weights and strengths of connections, neurons can enhance and weaken their interconnections, which allows them to learn to fire action potentials together, right? Send information together in synchronization, and thereby encode memories over many different timescales. And these properties of synaptic plasticity enable the brain to do memory storage and signal processing in the same locations. And that's quite a bit different from what we see in modern computers, where you have a CPU doing the processing and a memory core elsewhere doing the memory storage. All right, so we're now to the point of the presentation where you can close your eyes. Maybe you did that already, I don't know. But specifically, I want you to close your eyes, be still, and relax for a minute. Okay, so now that you've closed your eyes and relaxed, I want you to think about what you perceive with your senses. What do you hear? What do you smell? Maybe what do you feel? So keeping us moving, if we reflect on this, maybe let me ask you, did you hear or notice maybe white noise in the room, such as the air conditioning system or the heating system, or maybe the buzzing of lights? Did you hear traffic outside? Did you hear birds chirping? Did you smell your cologne or deodorant or your laundry detergent? Did you feel the socks and shoes on your feet or the clothes that you're wearing? Did you feel the chair in which you're sitting or the ground on which you're standing? There's a good chance that maybe these senses or these perceptions were not what you were aware of when I asked you to kind of think about what you perceive. But you were probably astutely aware of your clothing, for example, when you got dressed this morning, or the chair when you sat down in it at the first instant. So this simple example, this simple kind of activity shows that our senses not only allow us to detect things, but they readily adapt to their surroundings. All right, so now you need to open your eyes back up for the other slides. So this process is known as sensory adaptation, and it's defined as the reduction in sensitivity of a sensory receptor to a prolonged stimulus. So for example, the figure that I'm showing on this slide is taken from a paper by Delmas et al., and it shows the receptor potential of mechanoreceptors in rats, and it shows how these receptor potentials diminish over time in different amounts of time, depending on the type of receptor, under prolonged stimulus. And in this case, they were using indentation of the receptors. So these shrinking receptor responses lower the resulting frequency of action potentials that are then transmitted down the axon and to other neurons and potentially to the brain. So a biological advantage of this effect is that it allows neural systems to prioritize encoding of changes in stimuli, meaning new events that are sensed, rather than just continuously responding to persistent steady state values. And so in short, this is a way that nature saves attention and energy for things that are no longer new and interesting. So if we could replicate these types of properties, both the adaptability, the multifunctionality, think processing and memory in the same location, and the adaptive connectivity of biological nervous systems, then the field of neuromorphic engineering is primed to revolutionize how we compute and how we handle sensing in all types of systems. So neuromorphic systems that can process, learn, and remember are specifically primed for enabling large-scale computing, especially greater AI performance that's currently limited by our by traditional transistor-based technologies. But also, if these systems can emulate features of the nervous system that interweave sensing, signal processing, computing, and learning, then I think we can adapt and develop smarter technologies such as vehicles, UAVs, or automobiles, or autonomous robotic systems that can thrive and learn how to operate in challenging environments and maybe directly with people. And also even technologies that operate on or within our own person, such as wearable devices, brain-machine interfaces, prosthetics, or potentially biomedical implants. So in the past few years, research in my group at UT has been heavily inspired by these adaptive functionalities of neurons and synapses. Today, I'm going to discuss some of our work on kind of two lines of research in this space. First, I'll discuss our work to develop soft biomolecular artificial synapses, which we feel uniquely mimic the composition, structure, and signaling properties of neural synapses. This work was first sponsored by the National Science Foundation, and at the time it was really led by Professor Joseph Najim when he was a postdoc here at UT. And now it's being carried forward by some PhD students in my group, including Josh Mirage. Then I'll switch gears and discuss a recent collaborative project with Andres Arrieta's group in mechanical engineering at Purdue, in which we sought to develop reconfigurable material systems that integrate mechanosensing and learning capabilities. And so this work was sponsored by a DARPA Nature's Computer program grant, which was focused on exploring unconventional systems for enabling physical computing. So while there's been quite a bit of research in the last several years on a variety of devices that could be used to emulate activity-dependent plasticity found in synapses in the brain, one of the most common and most promising computing devices is called a memory resistor or memristor. So this is a two-terminal device, and one of it is sketched here, that exhibits voltage or current-induced changes in resistance. And so the figure on the right here in the graph illustrates maybe the potential current voltage relationship for this type of memristive response. And here we see that the device, which maybe starts in a highly resistive state, then is switched at a sufficient voltage level to a much more conductive state, what we'll call the low resistance state, or LRS. And so this, per this diagram, the switch occurs at a positive voltage, and then the device retains this more conductive state until the voltage becomes sufficiently negative, at which it can reset back to its original high resistance state. So this type of response would qualify the device as a memristor because we see pinched hysteresis in the current voltage plot. And the fact that this low resistance state is maintained even when the voltage goes to zero, right, when we take away the applied power, it qualifies the device as what's known as a non-volatile memristor. And so that means it can store permanently its change in resistance. So this can happen physically due to a number of physical mechanisms, and it usually incurs the formation of conductive filaments that are shown here in yellow between the outer electrodes of this two-terminal device. And the hysteresis in this current voltage plot reflects the fact that the present state of resistance of the device depends not only on the present value of the voltage, but also the history of applied voltage. And so this process is similar to how synapses can become more or less conductive as a result of incoming action potentials from the neurons. So the original theory for memristors actually was developed in the early 1970s by Leon Chua, but it was only first realized and confirmed experimentally by Stan Williams' group at HP Research Labs in the late 2000s. And there's some great reviews that discuss mechanisms and behaviors of memristors if you're interested. So these pinched hysteretic IV curves for memristors can come in what I call many different flavors, and here's some that are plotted. These were taken from a Chua paper. And the shape of the IV responses depends on the materials used and the mechanisms that allow the device to alter its resistance state. The fact that sufficient voltages can be used to change the device's resistance means that memristors, like synapses, also co-locate processing and memory. And that's an important kind of aspect of why they're promising for brain-like computing. So one of the ways in which a single device can then be scaled into a functional circuit or chip is to build what's known as a 2D, and there are even 3D variants of cross-point or crossbar arrays. And so these are architectures in which you have rows and columns of electrodes, and at each vertical intersection between these, there's a memristor device that can change its resistance based on the applied voltage. And in this sort of an arrangement, right, where you can easily see rows and columns of inputs and measured outputs, the vector of the currents that flow from all the columns of the array mathematically represents the dot product of the applied voltages and the conductances of all the memristors in the network. And so because of this, crossbar architectures enable very fast, very efficient analog vector matrix multiplication. And that's a core mathematical computational step in artificial neural network algorithms. And so this can enable really efficient, fast classification of static images as well as time varying signals. It's also useful in solving partial differential equations. So in most cases, and this image was taken from a review article in memristors, the conductances of the memristors are usually programmed in a way that emulates back propagation. So you will figure out what these conductances need to be and then apply voltages at the appropriate row column combination to tune the resistance of that node. However, depending on the type and arrangement of the memristors in the circuit, it turns out that these crossbars can even exhibit input-driven programming, meaning the applied inputs to the system that are being used during the processing part of the circuit can drive programming and concurrent learning at the same time. So the reference highlighted in yellow here was demonstrated by Joshua Yang's team at USC, and they showed a self-learning crossbar array for learning how to do pattern classification. I'm gonna close my Outlook real quick or keep dinging. Okay, so in engineered memristors, the increase in device conductance is often caused by voltage-induced movement of conductive species, and this can be metal ions or it could be oxygen vacancies. And so these species can move through either a polymer or oxide middle layer in the device. But the effect is that it results in kind of a local short circuit, which lowers the resistance as we looked at in graphs on prior slides. In contrast, in biology, cells use ion channels, such as shown here, to open and close and modulate the conductivity of ion flow into and between cells. So these are vital components in neural synapses, ion channels. And it turns out that most all sensory receptors in your body also use membrane ion channels to sense or transduce these external stimuli. So the fact that existing memristors, which are often hard to control and use due to low reproducibility and high write voltages and high conductances, that means high power consumption, voltage activated ion channels operate at low voltages, very small currents, and they're intrinsically nanoscale, and they have other advantages too. So the literature also reveals that existing memristors can only emulate a few types of synaptic functions. They ignore most of them, and so many types of kind of activity-dependent plasticity that is seen in nature is not passed on to neuromorphic engineering circuits. And I think this is an opportunity for new materials to provide a role in this space. So therefore, we proposed that natural and engineered ion channels could potentially provide a promising alternative for memristance and synaptic plasticity in neuromorphic systems. Our underlying hypothesis was that emulating the composition, structure, and transport properties of neural membranes, right, being more biomimetic, could yield new understanding and functionalities in modular, low-powered, brain-inspired synapses and neurons. And so by exploring biomolecules, which is vastly different from solid state materials and electronics, for doing signal processing and computing, this approach is different, but we think it could lead to soft, squishy, even multifunctional materials for sensing and computing. And you can imagine that this could enable vastly different types of application spaces, given the differences in material properties, length scale, and even the fact that these ion channels communicate via ions, not electrons. So in order to test our hypothesis over a couple projects, we've been researching the memristive properties of voltage-activated species in biomimetic lipid membranes, such as those that can be formed between lipid-coated water droplets in oil. And so the GIF or GIF on this slide shows the spontaneous adhesion of two lipid-coated water droplets that are submerged in oil. When they connect, which is an entropically driven process, and they connect spontaneously, the two opposing monolayers that cover each droplet kind of zip together almost to form a bilayer membrane at the interface that is very, very similar to a real cell membrane. And it allows us to then incorporate and study the activity of ion channel forming species. So we can incorporate these ion channel species, and these can be peptides or proteins or other small molecules. We can put them into the droplets and then look at their activity when they associate and interact in the interfacial membrane. To stimulate and interrogate this activity, we insert wire-type electrodes. This also allows us a way to control the droplet positions. And this approach kind of more broadly is used in a lot of biophysical studies looking at membrane behaviors and transport, and it's known as the droplet interface bilayer method. And it has some useful advantages, at least experimentally, for controlling and studying activity in these very thin 5 nanometer thick model membranes. So our research objectives for the work have been to explore memory and plasticity in voltage-dependent biomembranes, understand physical factors and mechanisms that can be used to tune switching dynamics and memory retention, try to understand useful signal processing and computing functionalities that these may provide, and then look at ways to assemble and characterize modular biomolecular neural networks. So scale them up to multiple elements to see what they can do collectively. We started really simple. And so as a baseline, we started by measuring the current voltage relationship of just a plain lipid membrane without ion channel forming species. So the plot on this slide shows the measured responses at 3 different voltage sweep rates, which are all very slow, relatively speaking. And that was done to minimize any capacitive current that could be incurred by these voltages. And that's because the membrane has an equivalent circuit, as shown here, that consists of a parallel R and C resistor and capacitor that describes the membrane. And then that's in series with a much smaller resistance value for the aqueous electrolyte on both sides of the bilayer. Note that R and C are both functions of the area of the bilayer, and C also depends on thickness. And so these quantities are dependent on the geometry, the nominal geometry of this contact between the 2 lipid-coated droplets. But the measurement shows that without ion channels, the membrane is a barrier to transport it at voltages below 200 millivolts. So we don't see very much current. This is on the order of 50 picoamps, so very small currents. We don't see any pinched hysteresis. So this shows that the lipid-only DIB is not a memristor. And this slight nonlinearity we can attribute to the fact that R and C change values a little bit when voltage is applied. So applied voltage can increase the area and reduce the thickness slightly. So in our baseline study, we saw that the lipid-only membrane is not a memristor. So now we wanted to incorporate voltage-activated species. And to start, we chose a model channel-forming peptide that's called alamethicin. It's made by a fungus, and it's an antibiotic used to kill bacteria. And from our own prior work, we knew that alamethicin peptides can be driven down into the membrane when an applied voltage is above approximately 100 millivolts. And when they're in the membrane, then they can group together to form these water-filled ion conducting channels. So the fact that channels form at a specific voltage threshold is similar to the, oops, go back, is similar to the voltage threshold dependent signaling that occurs in chemical synapses in the brain. The channel formation process is reversible too, and it can occur at both positive and negative voltages if these peptides are incorporated into both droplets, which would allow them to be present on both sides of the membrane. So when we did a similar voltage sweep and measured the current, we obtained traces like that are shown in this figure on the slide. So we see that the graph shows that the current voltage relationships measured on this lipid membrane decorated with alamethicin, initially at 0 millivolts, we see the ion current is negligible, indicating that the peptides are in a surface bound state. And then, however, when the voltage increases above about 100 millivolts, we see the current increases exponentially. And this shows that the channels have inserted and formed, or the peptides have inserted and formed ion channels. As the voltage reduces, these channels then exit the membrane back to the side where they came from. And this causes the membrane to revert back to a high resistance state. And the fact that this occurs before the voltage reaches 0 volts means that the device has volatile or transient membristance. So therefore, the membrane undergoes this resistive switching, and it exhibits short-term memory as reflected in the small amount of hysteresis here. And this response is symmetric because we incorporated alamethicin into both droplets, and it's present on both sides of the bilayer. The measurements also show at the different sweep rates that the hysteresis lobe area, kind of the area within these hysteretic curves, decreases as the sweep rate increases. And that just shows that the switching itself is not instantaneous. And that's a requirement in Chua's original memristor theory. There has to be some kind of characteristic time for the system to change its conductance forward and backward. So the next thing we did, my animations are out of sync here. We examined the response of alamethicin doped membranes to brief voltage pulses. And we did this to assess the dynamics of channel formation, meaning how long it takes for the channels to open and then close as a voltage that's above the threshold for insertion is applied. And as shown in the table, these measurements revealed that the time constants for switching, which we characterize in kind of 3 ways, are on the order of 10 to 100 milliseconds mostly. And we could tune these a little bit by different lipids used to form the membrane or by changing the temperature of the system. Interestingly, they're on the same order of time constants that are seen for neurotransmitter activated ion channels in the brain. So applying then many subsequent pulses, not just one, caused cumulative changes in the conductance. And so you can see that in this figure of current versus time, where we applied a pulse train of rectangular voltage pulses. And it shows that the membrane and its channels plastically adapt from the remembered prior states. And so changing the time between the pulses then influences whether the conductance accumulates or rises, and this is sometimes called paired pulse facilitation, or whether it reduces. And so shorter times between pulses allow the memory of the prior state to be better retained, and so the device facilitates its conductance, whereas longer times between pulses allows it to forget more of that prior activity, and you get a weaker response pulse to pulse. But these are very similar to what's seen in short-term plasticity in synapses. We were very excited by these results because this was the first time, to our knowledge, that people had studied and demonstrated membristance in biomolecular ion channels. But we wanted to understand the mechanisms that drive these changes. And so as engineers, we also wanted to model, we wanted to develop a model to describe these dynamics from our prior work on these droplet interface bilayers and alamethicin, we knew that the applied voltage can have two effects. It can cause ion channel formation, which increases conductance, and it can increase the area of contact between the droplets through a process known as electrowetting. So with these in mind, we developed a simple model for our biomolecular memristor, and that's kind of summarized here, which relates the current through the device to both the applied voltage and the conductance of the membrane, which depends on how many channels per unit area, how many ion channels per unit area are present, and what the area of the membrane is. So then using either pre-published dynamic expressions for the channel formation rates for alamethicin or empirical expressions for the dynamics of electrowetting, we're able to come up with an overall expression for the conductance of the channel. And then what we did, we knew some of the terms in these expressions, either by direct observation, but in some cases, we had to fit the data to the model to determine unknown parameters, such as some of these terms in the dynamic expression for alamethicin channel formation. But this allowed us to get a model that, that described and helped reveal the key sources of plasticity and memory in our devices. So this procedure revealed fundamental differences in the rates of channel formation and area change due to electrowetting. And to illustrate this, we simulated the response of our biomolecular synapse to a series of rectangular pulses, kind of shown here. And with time constants on the order of milliseconds, we see that the simulated number of conductive channels per unit area rapidly increases and decreases with each cycle of the pulse train. And then after several cycles, it kind of reaches a steady state dynamic behavior. In contrast, the voltage waveform, again, this rectangular pulse train, caused a much slower exponential rise in membrane area that took several seconds because those physics are slowed by the viscosity of the liquid environment. So since the total number of channels is the product of channels per unit area and the change in area, then we could simulate the change in current versus time and show that with the right time between pulses, you could get these kind of monotonically facilitating changes in conductance across many cycles. And again, the current shows that shows the individual effect of channels opening and closing with each pulse, but also the overall increase in the number of channels across many pulses. So what we were able to conclude from this study is that the slow increase in bilayer area and fast insertion of new channels leads to short-term facilitation responses. And these looked a lot like what we measured in our experiments. And that the hysteresis in the IV relationship, right, the hysteretic IV curves, is primarily due to the time lag for the area growth to add new channels. So a bigger area can incorporate or host more channels, as opposed to the channel hysteresis, which was caused by channel relaxation. So in follow-up, then we published a couple other papers that built on these discoveries, and one was also led by Joseph, in which we looked at the dynamic changes in capacitance versus voltage and demonstrated a property known as memory capacitance. And then my former student, Shubhadeep Koner, demonstrated behaviors that leverage these physics to mimic artificial electrical synapses, which are formed by gap junctions between neurons. Using our model, though, we also collaborated with folks in electrical engineering at UT to explore whether the properties of biomolecular synapses provide any computing advantage, right? Are they useful? That's a basic question. So here, in order to kind of evaluate this, they performed evolutionary optimization to configure hidden neural networks in which the synapses between neurons were defined using our model for the alamethicin memristors. And then the models were evaluated in tasks such as classification of images or classification of temporal signals. So this schematic shows the optimal network layout for a hidden neural network for doing EEG signal classification, and it's used to detect epileptic seizures or signatures of epileptic seizures. The network contains only 6 neurons and 6 synapses, and it demonstrated greater than 97% accuracy in distinguishing these features. And really interestingly, it didn't require any hidden neurons. And the model requires, or the network requires, significantly fewer synapses and neurons than as compared to networks simulated for solid state memristors. So there's something interesting there in finding that by using the properties of these biomolecular synapses, which in some ways better mimic signal plastic or activity dependent plasticity in synapses, that we maybe get more efficient computing and signal processing. More recently, my student Josh has been studying memristance and plasticity in membranes doped with different species, and one of these is called monosmycin. And kind of the hallmark difference compared to L-methicin is that it's a 3-state species. And so it can reside in what we call a non-conductive pre-channel state on the first side of the membrane, a transmembrane conductive state that transports ions, and then an inactive non-conductive state on the opposite face of the bilayer. So shown here are the current voltage relationships for monosmycin doped membranes. And like L-methicin, we see that these show pinched hysteresis, and that channel formation occurs at similar voltages, around 100 millivolts. And we see, based on kind of the counterclockwise path of current in both the first quadrant and the third quadrant, I didn't include the arrows here, it confirms that the device turns off before the voltage reaches 0, and that shows that these also are volatile memristors. So the hysteresis, or the memory, is lost when the voltage is taken away. So the fact that I'm plotting current as a current density shows that when we normalized by the dynamic area of the bilayer, we could see just how the current through the channels changes versus voltage and time. And so this proves that the channels themselves have the memory. It's not just electrowetting like we saw with L-methicin. And so this was an exciting result. The effect of the inactivation was more prevalent when we did a different type of measurement. So here we applied a step voltage of different amplitudes and then measured the current through the membrane at each of these amplitudes. And we see that the amount of current through the membrane and the shape of these dynamic responses depends on this amplitude of the step. Specifically at lower voltages, it's kind of an S-shaped saturation response. And so we identify this as kind of a facilitation only type response. And it leads to a steady current at steady state. At higher voltages, however, the channels are quickly pushed into the membrane, make it more conductive, and then it begins to inactivate. And the device, even though the voltage step is still on, the device starts losing conductance over time. And so this reflects the fact that the monosmycin channels are being pushed across and out of the bilayer on the opposite side. So we termed this response facilitation then depression, and we equate it or compare it to sensory adaptation that we thought about a few minutes ago. So for this type of response, we also developed a 3-state kinetic model to capture and really explain some features of this nonlinear dynamical response. And this was published in a paper in 2021 that provides more detail on the modeling. The last thing we did here was to demonstrate this bidirectional type of plasticity is in fact a form of sensory adaptation, or could be used as such. So we constructed and tested what we call a hybrid afferent synapse, which consisted of a photosensor and 2 transistor-based leaky integrate and fire neurons that output spikes when they integrate current. And then between them, we included our monosmycin biomolecular memristor, or biomolecular synapse. And this arrangement kind of mimics the the functionality and organization of the retinal neurons. In that light is detected, it excites sensory neurons, which then through their synapses adapt and transmit this information to the visual cortex. In our system, the amount of light and thus the current output by this photodiode controlled the spiking frequency of the neuron, of the first neuron. And so when the time between the pulses, which we could tune, was appropriate, the RMS voltage difference across the synapse, right, the RMS voltage difference between the output of neuron 1 and the input of neuron 2 caused channels in the membrane to insert and become conductive and exhibit their dynamics that we looked at on the prior slide. And this subsequently controlled the amount of current that flowed into the second neuron and determined how often it would output a spike upon integrating that current. And so what we see in the graph here is that the 3 levels of light we tested resulted in different frequencies of neuron 1 firing. But then these signals, which effectively become RMS voltages across the memristor, then create different types of dynamic responses measured as current through the memristor or as frequency of spiking of neuron 2. So at low light levels, current in the synapse, and neuron 2 facilitated and saturated, while at higher stimulus levels, higher light levels, we see both signals, current and spiking frequency. They facilitate rapidly and then they depress with this continued stimulation. So this shows that the synapse adapts to the continuous stimulation in a way to reduce the amount of information sent upstream to neuron 2. And this was also part of Josh's paper. So in part 2, I'm going to switch gears. I'll introduce work that we did on a recent project funded within the Nature's computer program, which was sponsored by DARPA. And this program sought innovative research concepts, I'm borrowing some language from their website, that exploit the interplay between dynamic behaviors and intrinsic material properties for enabling new forms of embedded physical computing. And so our specific project, which we did in collaboration with Andre Sarrietta's group at Purdue, examined mechanosensing and associative learning on flexible substrates that we called neuromorphic metamaterials. All right, so the physical basis for our approach was this new type of multi-stable reconfigurable mechanical metasheet shown in the figure. And I also have one here. So it's like these little domed sheets that are flexible, and each dome can be popped in and out. And so these structures consist of soft sheets featuring a patterned array of bistable domes. And kind of like the Poppets kids toy, the domes can be reversibly inverted at the local scale, but kind of unlike that toy, when you invert certain domes, then it causes, can't find it, it causes the overall sheet, the meta sheet, to undergo some overall shape change. And so this results in programmable multi-stable shapes of the sheet, and some of these are shown here, and even tunable mechanical properties, stiffness values and curvature, et cetera, at the global scale. So in short, what's interesting about these types of meta sheets is that the final configuration of sheet depends on which domes are inverted, but also in the order in which they're inverted. So you might arrive at different final states depending on which order you pop certain domes. And it's this nonlinearity between these applied forces that invert the domes and the final shape that we thought provided a compelling basis for computing, meaning classifying patterns of applied forces based on the resulting induced reconfigurations. So there have been several examples in recent literature the last couple of years where people have looked at neuromorphic materials that integrate with sensory systems to encode and process external inputs to do online learning, which means the sensory signals cause adaptation kind of in situ, and that affects the resulting processing. And this is a crucial function of neuromechanical systems that has really been much less explored. So while they've been done, this functionality of online learning in neuromechanical systems is less studied. Moreover, engineered material systems that integrate mechanosensing, memory, and autonomic learning capabilities are pretty rare. So in order to retrieve information about the conformation of the sheet, right, if you imagine applying forces to your sheet and then observing its reconfiguration, the state of the system has to be transduced, and it has to be done at the local dome level. So what we proposed was to incorporate resistive strain sensors that were printed into the meta sheets, and we would connect these electrically to resistive memory elements, memristors, to be able to provide a sensory reception and memory storage capability. And our hypothesis was that by reading the final electrical states of the memristors, we could retrieve the patterns of the applied forces that were applied to the domes. So first, Andres's team printed these domes, and they printed conductive resistive strain sensors around this. I'm sorry, it's a little small to see this. You can see that when the dome's inverted, the curvature of even the base sheet around the dome is changed, and that's what's being sensed by the strain sensor. And they measured reliable changes in the resistance as measured from these resistive strips in the sensor. At the ground state where the dome's not popped, the resistance is low. At the inverted state where it's popped, the resistance is high. And there's a transient that occurs during the popping event. I think it briefly loses conductivity. But these changes were on the order of several hundred ohms. And so this provided us confidence that we could locally and reliably sense dome inversions using these printed strain sensors. Then what we needed to do was store these inversion events in the electrical domain. And so we needed memory elements that could communicate with the sensors. And in this project, we weren't using our biomembranes. We rather developed and tested thin film memristors, which were made with a conductive polymer P.PSS, which was sandwiched between a copper electrode and an ITO conductive substrate. We chose this polymer based device for the purpose of eventually enabling these memristors to be incorporated directly onto the meta sheets. For our initial study, they were off the sheet, but electrically connected to the sensors. So as shown on the right, this behavior yielded fairly stable resistive switching that occurred at voltages of about 3 volts. And they exhibited kind of non-linear but non-volatile memristance for dozens up to hundreds of cycles. And so this provided the necessary proof of concept for showing that we could store electrical memories as resistive states. So we then developed a scheme to integrate these, and this required biasing electrically the sensor, connecting that through an amplification regulation circuit that helped control a voltage that was then shared across a series resistor in the memristor. And in doing this, then the current through there creates a voltage divider, and we can monitor the voltage across the memristor using a simple Arduino circuit, and then track changes here as measures of events that happened on the dome. And so we tested this ability, and one of the first things we wanted to understand was if we applied incremental or discrete voltage pulses. You can think about it as VN signals, which would replicate or represent dome inversion events. Would the memristance change in an incremental fashion? And so the data showed here shows that yes, indeed, the memristance does change, and we would always read the resistance between pulses at a voltage of about 1 volt. And so we found that a 5-second pulse or voltage pulse, or a 5-second dome inversion, so to speak, created the most consistent and the largest cumulative change in memristance versus pulses. So then the challenge was to try to demonstrate that we could somehow learn patterns in this sheet. And for doing this, Andres's team really led this part of the work, but they instituted what's known as a Hopfield network. And Hopfield networks are they're a type of associative memory network, which can store different memories, and it's stored as attractors or fixed points in some numerical energy landscape. And these can then be retrieved via a post-processing energy minimization calculation. So in our prototype, we devised the physical hop field network, which consists of N by M dome arrays, each with their own sensor, and and what we did was we used the sensors and the difference in the changes of the sensor resistances to then program 6 memristors. And this allowed us to create, to store these kind of interaction parameters, or the differences in resistance, store them into the top, the upper right-hand triangle of a interaction matrix, which is called the J matrix. And it's this matrix that undergoes an energy minimization calculation to determine what were the patterns that were applied to this to allow the system to reach its final state. So in order to do this, we would test devices. Let's see if I can get this to play. And so this is a video showing my students popping devices and then kind of subsequently measuring the changes in voltage across the memristor. And this corresponds to changes in memristance, if you might recall. And so through a scheme which involved kind of initial calibration and then doing the training where we would write these changes with the imposed dome inversion patterns, then we could look at differences between the final state of the memristor and its initial state to essentially count how many times was that dome inverted. So I'm going to continue on. And what we tested was the ability to kind of program or train multiple patterns in a sequence into the final state of memristance that was used to form this interaction matrix. And without going into the math at all, we could then extract these energy landscapes to find these energy minima. And these correspond to the different patterns that are applied to the sheet in order to achieve this final interaction matrix. And in short, again, the team at Purdue did this part, so I'm kind of naive in discussing the details. In short, we found good accuracy above 90% in matching, in retrieving the pattern of dome inversions that were applied as compared to what was predicted or what was extracted from the data. So I'm just going to summarize really quickly here. We kind of talked about 3 areas of research. The first is just that biology achieves efficient computing and signal processing by leveraging stimuli responsive and activity dependent adaptations in neurons and synapses. We've tried to look at that both in biomolecular synapses and even engineered polymeric materials. And if it's done well, then I envision a future of enabling these technologies for more efficient AI platforms and also driving the development of smarter autonomous engineering systems such as robots. And there's some relevant challenges with each part of these research fields. So what's next? Well, one of the things that's at least next in my group is that we're trying to develop neuromorphic biomimetic interfaces that can actually function at the edge of biology, which is kind of my term for thinking about near or in contact with living tissues and cells. And so the goal of this research, which is a new project funded by the AFOSR, is trying to combine the materials and synaptic-like properties of organic electronics, stimuli responsive proteins and biomolecules, and even traditional inorganic and hybrid electronic circuits to develop these kind of neuromorphic interfaces that can help bridge the gap between nature and traditional electronics. And so this is new work in collaboration with Jonathan Rivney and Neha Kamat at Northwestern and Yong Chen at UCLA. And I selfishly, I'm advertising for a postdoc that I'd like to have joined to work on this project. And so if you're interested in brain-inspired biomolecular electronics, or you know someone that may be, please have them email me. I'd be happy to chat more about the project. So with that, I'll just quickly acknowledge the funding for the work that I showed you today, and definitely like to thank the people that did the hard work, including Joseph, some of that early work, and some of our collaborators. So I hope I've left enough time for a few questions at this point.
Yeah, we have 3 questions. This was a great talk, Andy. Thank you so much. Let's go ahead and start with question number 1. Can you see the questions, Andy?
I'm
They're
trying to
Q&A.
organize all my windows. Let's see. So what parameters of biomolecular synapses are adaptable and learnable? That's a great question. So I zoomed through it just in the interest of time. What happens in synapses, these are where neurotransmitters are released and picked up by the 2 neurons. And so the ability to communicate information depends on how many neurotransmitters you can release and how many of those chemical signals you can detect. And so during plasticity and adaptation, the number of receptors at these junctions can be increased up or down. Even the chemical states of these receptors can be changed through phosphorylation processes. But also the morphology and the size and structure of the synapse can be changed over time. So the cell can adapt its morphology to build stronger or weaker connections. So it's actually multiple mechanisms that drive these different forms of plasticity. Hope that answers the question.
So we have another question from Ahmed Muhammad. I don't know if you can see that.
Yeah. So for the droplet interface bilayer neuromorphic devices, you mentioned that their lifetime usually ranges from hours to days. Doesn't that pose a challenge in building devices with large numbers of devices? Yes. Do you see a path in terms of improving the mechanical stability of such a liquid-based technology? Perfect question, and you're exactly right, especially if you've ever looked at these in the lab in person. These have provided a lot of value for controlling the experimental platform to understand the basic aspects of voltage activated channels and the properties that we think we could get. But I don't think it's feasible to, at least in the short term, build systems that are stable based on droplets, right? It's more of a salad dressing than a device. And so our new project is seeking to overcome that in part by using organic electronics, which are similar to hydrogels, depending on the material you use, and cushioning biomembranes directly on these surfaces and then encapsulating them in ways that I think could enable better durability, longer lifetimes, and also build larger networks of devices for operation. So I think that that's one pathway forward.
All right, we have another question from Nicholas Armendariz.
Yeah, so are the copper PDOT PSS ITO memristors well suited to neuromorphic tasks shown by the biomolecular memristors, or do they lack some synaptic qualities? That's a great question, Nicholas. So what we've seen, what we needed for the DARPA project was a memristor that could retain these changes in resistance. So the ones that we exhibited had non-volatile memory, they were more permanent, whereas our biomolecular synapses could exhibit memory, but only on the order of milliseconds to seconds. So they would lose these histories. Some of the complex adaptability, like the facilitation then depression or the sensory adaptation, we have not seen in the PDOT-based memristors. So there's maybe a loss of some functionalities there. I will say just kind of really quickly that my student Shuv is working on a paper that describes how these PDOT-based systems work, because they're made differently from what we've seen in the literature. And he did a study where he replaced the other ITO electrode or the ITO electrode with another copper electrode. And when he made the device symmetric, then we actually reverted from non-volatile memory to volatile memory. And so we think that that could be a way to kind of bridge these types of properties. I hope that that starts to answer the question you had there.
I think you answered it great. So I think we're at 12 o'clock. So thank you so much for participating in our seminar series. I think this is, this is very close to heart to me. This is very exciting work. And for the future, for our audience, we have our next LIM talks at the end of November 30. We'll announce the speaker soon, so hope to see you all soon. And thank you all. Zubaida, you want to say something?
I just want to really thank Angie. I loved your seminar. It was awesome. And I'm very excited by what you presented on the dome metastructures. I do have a quick question. It looks like you are focusing on the mechanosensing, and that makes sense, right? In terms of relating it to what you've done before. What about, I see a lot of potential in terms of actuation. I was jumping up my seat. And especially, you know, thinking about the origami-like inspirations. Is that something you or anybody else is working on?
I think Andres is leading some of those efforts. So he makes these sheets. And again, like I sit here while I was talking, I was playing with this little domed sheet.
It's so relaxing, right? It's
my kids like poppets, so it's like poppets on steroids in a way. But you're exactly right that, you know, you get a shape change. So if you can cause these inversions, then you can actuate the structure. Some of the initial things that they've looked at is actually encapsulating the back and being able to control pressure to locally pop in and pop out. And they have, I don't know if I put the citation there, they have a paper where they demonstrated some simple kind of robotic gripping type functionality with these dome sheets. The mechanics are really interesting. And I think, and they're really rich for understanding how you can have these kind of coupled mechanical responses across local length scales and global length scales. I also think on the sensing side that there's probably other ways to retrieve information about the state of the system. So it's definitely something that he and I want to keep pushing forward. Our initial paper or study, I think, showed some good proof of concept, but we have some ideas on how it could be more useful.
Very cool. I'll look that up. Thank you so much.
Yeah, it's good to see you. Thank you both for the invitation. This was great.
Very welcome. All right, everyone. See you next month. Bye-bye.
Take care. Thanks, everyone.
Bye.
Bye.